Unit 1 covers 6 topics, but underneath all of them, it's only making 2 claims. 2 sentences. Get these, and the whole unit stops being 6 separate things to memorize. So let's fill them in, one molecule at a time. Start with the question all 6 topics are quietly asking. What makes a molecule that molecule? Here's glucose. Here's fructose. Same formula. 6 carbons, 12 hydrogens, 6 oxygens. Identical formulas. So, same molecule? No. One closes into a 6 ring, the other a 5. Same parts, different arrangement. And they behave differently. Same atoms, different molecule, different job. So a formula doesn't name a molecule. That's the first crack. One level up. 2 molecules. Exact same carbon backbone. Atom for atom. Change one thing. The group on the end. Acid left, amine right. Now one's an acid, one's a base. They react with each other. Opposite chemistry. The backbone is scaffolding. What it does is decided by what's attached. One more level. Monomers link into polymers. Squeeze out of water, snap a bond. Now thread the same 4 monomers in a different order. Same inventory, same bonds. Completely different molecule, because order is arrangement. 20 amino acids, a chain of 100. More orders than atoms in the universe. So, sentence 1. What a molecule isn't what it's made of. It's how it's arranged. Now sentence 2. Back to the first molecule of the unit, water. 2 waters. They stick to each other. This dashed line is the hydrogen bond. Not the bond inside the molecule. That's covalent, far stronger. The hydrogen bond is the dashed one, between molecules. Boil water and only the dashed bonds break. The molecules leave whole. Steam is still H2O. So keep a ledger. Strong bonds, weak bonds. Heat only reaches the weak lane. Strong bonds build the thing. Weak bonds arrange it. 2 molecules side by side. A protein on the left, DNA on the right. You've seen both. 10 amino acids, 9 peptide bonds. 10 nucleotides, 9 backbone bonds. Same number. Hold on to it. Both of those are covalent. Both are the strong lane. They are what makes each one a chain. And holding each one in shape? Dashed lines. Both of them. Hydrogen bonds folding the protein, hydrogen bonds pairing the bases. So heat them, both at the same time. The protein unravels, the DNA unzips. Both shapes are gone. Both stop working. Now look at the counters. 9 and 9. Not one covalent bond broke, because heat never had the strength. A hydrogen bond is 20. A peptide bond is 350. Heat takes the shape and leaves the chain. Shape lost, sequence kept. That's the definition of denaturing. And it's the same sentence for both molecules. There's sentence 2. And since the chains survived, cool them down. Both find their shape again. That's not a cartoon. Heating and recooling DNA is exactly how PCR works. Here's the quiet miracle. That sequence folds the same way every time. Order decides the fold. So change 1. 1 amino acid out of 146, charged to oily. That's sickle cell. No bond broken, 1 letter, a different shape, a different life. There's the hinge. Arrangement is the instruction. The weak bonds carry it out, which is why the unit keeps saying it. Sequence sets shape, shape sets the job. So, the 6 topics. Elements, macromolecules, properties. All sentence A. Skeleton versus groups. Order, isomers. Water, protein structure. Nucleic acids. All sentence B. The same weak bond 3 times. It really is the same bond. The line you met between 2 waters folds your proteins and pairs your bases. Arrangement is identity. Strong bonds build, weak bonds shape. Denature anything, and the chain survives. 6 topics, 2 sentences. Step through any of them yourself. Links below.